Welcome, dear students. Today I'm going to explain some of the practical aspects of the core paper 1, that is phycology and microbiology. And in this class, today I'm going to explain some of the basic and fundamental aspects of algae, and today I'm providing you some of the crucial, most important features of Nostoc. From the previous classes, we must have a clear idea about what algae is. I think we all can recall that these are chlorophyll-bearing autophytic members of thallophytes, and there are so many types of algae. They form various forms and various types of thallus organization. Already I discussed in my previous class on the thallus architecture, and today simply we study the vegetative and reproductive structure of Nostoc. We all know that Nostoc belongs to the family Nostocaceae, order Nostocalis, class Cyanophyceae, and the division Cyanophyta. So, Nostoc, Anabina, Oscillatoria, Gliotrichia, Stigonyma, Stigonymata, etc. All these are member of BGA or blue-green algae. Now, according to the syllabus provided by the West Bengal State University, we have to understand the basic vegetative and reproductive structure of Nostoc. Firstly, I want to mention that these are freshwater or terrestrial algae and also found abundantly in paddy fields, and some of the terrestrial one grow on moist soil mixed with other plants like mosses, lichens, etc. And in some cases, they are also found as independent one, and in some cases, they also occur as endophytes with some other plants. Like they may be associated with some plants, and also they may develop as a predominant algal representative in the rice field. Now, if we want to study their vegetative structures or vegetative body, they form a filamentous structure. The filaments are aggregated together within mucilage enveloped, forming a ball-like gelatinous colonies, usually known as Nostoc balls, and they vary greatly in size and shape. The filaments are unbranched. They are usually contorted and twisted in various ways. Now, if we study these filaments under the microscope, It shows in a filament all the cells are not alike, but uh, that at interval are found slightly larger, rounded, and thick-walled cells having transparent contents. These are heterocysts. The heterocysts are commonly with two polar nodules, but rarely with one polar nodule also. And besides the formation of heterocysts, the filaments may form the specialized structure known as akinets. So in this picture, I think it is very much clear these are akinets, and this is the heterocyst. And in the two pole of this heterocyst, they form polar nodules. And this is a typical Nostoc ball, and portion of the filaments seen under the microscope after collecting from the original sample of the Nostoc ball. And this is one single filament with a trichome. Under light microscope, if we observe a live specimen under high resolution, they show like this, and the arrow indicate the location of the heterocyst. A heterocyst usually they show their intercalary position, and in some cases they also show their terminal position also. Now, reproductive nature or reproduction of this genus. Usually they follow the mode of fragmentation of the filaments into hormogones, and each hormogone on germination develop into a new plants. And in other cases, they also reproduce by akinets or spores. In some rare instances, heterocysts may act as spores, but usually the heterocysts they help in nitrogen fixation. In this next picture, this is the stages and germination of the heterocyst. A heterocyst, these are two polar nodules. Their division, there's subsequent division. And this is the germination of hormogone, what I already mentioned, to form a new colony. Thank you.